So, good afternoon. I'll discuss some therapeutic targets which have been identified to treat cardiac muscle disease. And as an example, I will highlight the observations in hypertrophic cardiomyopathy. So, hypertrophic cardiomyopathy is a disease of the heart muscle. And what is seen in the clinic is that there is this asymmetric remodeling of the heart, hypertrophy most of the time of the septum of the left ventricle. And if you zoom in at a histological level, you will see myocyte disarray and fibrosis. And the main dysfunction seen is impaired relaxation, so diastolic dysfunction. So people with this disease do not have an impaired systolic function, but show impaired relaxation. Now, genetic studies have shown that the cause of the disease is often in the sarcomeres. So mutations, heterozygous mutations affecting only one allele, have been found mostly in genes encoding sarcomeres. And the most frequently affected genes are myosin heavy chain, myosin binding protein C, and troponin T. So if you think about the development of this heart muscle disease, starting with a mutation in one of the alleles, and then thinking about disease progression, we now think that with aging, there's a shift from relatively small level of mutant protein towards an increase in the level of mutant protein in the heart. And so a mutation will alter sarcomere function, and starting as basically a mutation carrier without symptoms and without remodeling of the heart, many of these mutation carriers end up having this hypertrophic cardiomyopathy. Now, many studies have been performed in patients in the clinic, and as I indicated, the most striking function impairment is a diastolic dysfunction. And this has been observed already at an early stage of the disease development. So in people who do not have advanced remodeling, who do not show hypertrophy of the left ventricle, impairment of relaxation has been observed. Now, another striking observation is the study performed by the team in Oxford, where they showed a reduction in the phosphocreatine ATP ratio. And this is an indication of the energetic status of the heart. And all patients showed a drop in the phosphocreatine ATP ratio. And the term energy depletion was introduced then because it is thought that the sarcomere mutations cause very inefficient sarcomere contraction, leading to inefficient contraction of the heart. Now, our own team has performed many studies using PET MRI imaging in mutation carriers. So these are the green symbols in this image, and also in people with advanced hypertrophic cardiomyopathy. And in all these people, we find a reduction in the myocardial efficiency. And this is the definition of how much oxygen is consumed to produce or for force development by the heart. So the dotted line is basically the levels which we find in healthy control persons. And you can see that most of the people who carry a mutation, the preclinical mutation carriers and also the HCM patients, have a reduction in cardiac efficiency. So from the patient studies, we see that there is diastolic dysfunction, and there's this impaired energetic status of the heart. Now, if you then look at the cellular pathomechanisms which may underlie these changes, I'll highlight three which are very well established based on studies using proteins, using animal models, and also looking at human cardiac tissue. And I'll pass these 3 changes. So myofilament calcium sensitivity, the myofilament kinetics and tension cost, so how much ATP is consumed, and the status of the myosin heads. Now, one of the first observations when people started to look at the effects of mutations which cause hypertrophic cardiomyopathy is this increase in calcium sensitivity. So if you would look at a wild type or control protein, this would be a normal dose response curve of the myofilaments to calcium. So at relatively high calcium concentrations, there's force development, which would be linked to the systolic phase of the heart. And when you have low calcium concentrations, as observed during the diastolic phase, force development is relatively low. But with a sarcomere mutation, this force calcium curve is shifted to the left, and this we call an increased calcium sensitivity, meaning that there's a high force development both at relatively high calcium concentrations, but also at relatively low calcium concentrations. Force development is still high. So this would impair relaxation. This could cause the impaired, this could explain diastolic dysfunction, but it would also mean that at any given calcium concentration, the ATP consumption of the sarcomeres, of the myofilaments, would be high. Now, what we've done in a study using human cardiomyocytes is sort of try to link, figure out if the location of a specific mutation can explain the change in calcium sensitivity, or if the dose of the mutation is of any relevance. So you can think of choosing different mutations depending on the location in the gene. And also you could think that the mutation protein dose would play a role in changing calcium sensitivity. So a low dose may explain mild symptoms, and a high dose could be linked with more severe symptoms. So we used isolated cardiac muscle cells, and we can do exchange experiments where we basically give the mutant protein as in the incubation medium, and then there's a competition with the endogenous troponin present, and you can do an exchange experiment. Basically, the healthy protein is kicked out, and the mutant protein is entering the cardiac filaments. And you can use different concentrations, so you can mimic a low dose and a high dose using this system, and also compare different mutations. So look at the mutation location and at the level of the mutant protein in the sarcomeres. Now, using this system, and Maaike Schult was the PhD who performed these studies, and what she found is that for a specific mutation, it did not matter if there was a low dose or a high dose of this specific mutation. They both increase calcium sensitivity to a similar extent. Basically, already at the low dose of mutant, there's an increase in calcium sensitivity. Now, if you then compare the different mutations and the effect on this calcium sensitivity, we did find a difference. So depending on the location of the mutation in the gene, in the protein, the magnitude of the shift in the calcium sensitivity was different. So one was more, provided a larger increase in calcium sensitivity than the other one. And this is in line with studies which have been performed by colleagues in Italy in mouse models. And basically, overall, the overall message is that really there's a specific mutation effect when you look at troponin T mutations in this case on disease remodeling, on the pathogenesis, which is seen leading to hypertrophic cardiomyopathy. Now, I would like to highlight that in addition to the mutation-mediated increase in calcium sensitivity, based on many studies, we also know that many other post-translational modifications cause an increase in calcium sensitivity. So we know that low PKA-mediated phosphorylation shifts this curve, and this is linked to a deregulation of the beta-adrenergic signaling in the heart. Oxidative stress, which may develop during cardiac disease development, also alters the myofilaments, and mitochondrial changes, which may increase the levels of ADP, increase calcium sensitivities. And we believe that these occur during disease development, but are secondary to the mutation-induced changes in calcium sensitivity. Now, the second cellular mechanism, which I want to briefly highlight, is the change in kinetics and tension course. So we've performed studies in collaboration with the team of Corrado Pugliese, where Corrado has this beautiful setup to measure kinetics of activation and relaxation. Basically, you look at the binding of myosin and the detachment of myosin heads to the thin actin filament. And for any cycle, ATP is consumed. So if you have faster kinetics of activation and relaxation, it's more ATP is consumed. And this is a troponin T mutation, which was studied in comparison to myofibrils from non-failing donor hearts. And what was observed is that the kinetics of activation, but also of relaxation, are much higher for the mutant sample compared to the non-failing sample. So there's an increase in kinetics of myofilament kinetics. And we could also measure ATP consumption using this setup, where we measure force development and at the same time the ATP utilization, and we can calculate tension cost. And this means that how costly is the development of tension, so how much ATP is consumed for the development of force. And for all cardiac samples which we've studied, carrying a myosin binding protein C mutation, a myosin heavy chain mutation, or a troponin T mutation, they all show a higher tension cost compared to samples where there's no mutation. So the control group here is the sarcomere mutation negative group. So there's more ATP consumption if there is a sarcomere mutation present in the myofilaments. And then the third mechanism, which has received a lot of attention in past years, is the so-called super relaxed state of the myosins. So we know, based on many beautiful studies performed in skeletal muscle, but also in cardiac muscle, that there's a portion of myosin heads in the so-called super relaxed state, which is an energy conserving state. And this was a study performed by James McNamara together with Christos Remedios, where they showed that there's a reduction of the number of myosin heads in the super relaxed state if there is a mutation in myosin binding protein C. And this means that basically there's an increased ATP turnover, as there's a lower number of myosin heads in this energy conserving state. And similar observations have been published for myosin heavy chain mutations. Now there's this recently, has been this trial using mavacamten, and many studies performed by the team of Christine Seidman have shown that mavacamten is able to shift this myosin status from a disordered state towards the super relaxed state. So as I indicated, in the hypertrophic cardiomyopathy samples, there's a change from the super relaxed state towards the disordered state. And mavacamten is able to reverse this, or to switch back to the super relaxed state, which is more energy efficient. So less energy is used in this state. And so this mavacamten was already tested in a clinical trial, which was published in the Lancet, led by Jacopo Olivotto. And as indicated, the mechanism of action of mavacamten is basically targeting these myosin head, shifting it towards the super relaxed state, and thereby improving myocardial energetics. At least that's the idea behind it. And the results of this intervention were very positive. And I'll show just briefly that the output gradient, so the left ventricular outflow track gradient, which is increased in these patients, was highly reduced with the mavacamten treatment compared to placebo-treated patients. But the overall mavacamten had an enormously positive effect on NIA class, improved exercise performance, and also reduced biomarkers in the blood, which are linked with impaired cardiac function. So those are promising, that's a promising results, having a drug targeting a mutation-mediated defect. But what was striking is that in this trial, there were also many people who are not sarcomere positive. And that's something striking of this whole group of patients, that 50% of the patients with hypertrophic cardiomyopathy actually do not carry a sarcomere mutation. So basically, the exact cause of the disease is unknown. So to figure out if we could tease out what's the disease mechanism in people without a sarcomere mutation, but also with the different genes affected, we performed a proteomics analysis and in a large number of samples. And what's obvious is that the hypertrophic cardiomyopathy samples separate very well from the non-filling donor samples. But if you look at the genotype, there's no distinction between the samples. And also, if you look at the sarcomere mutation negative group, which are the blue symbols, they also overlap with the sarcomere mutation positive groups. So cluster analysis does not separate them based on genotype. So we did look further which protein clusters are altered, and one cluster really got our attention, and that was the microtubule cluster, which showed upregulation of proteins. And why were we triggered by this? Well, the team of Ben Prosser performed many beautiful studies and showed that a change in microtubules, and more specifically an increase in microtubule detyrosination, stiffens cardiomyocytes and impairs contraction and relaxation of the heart. So I won't go into details because of the time, but this really is a new target in the cardiomyocyte, which is changed during heart failure, and which may explain impaired contraction and impaired relaxation of the heart muscle. So we went on and we performed a lot of Western blot analysis to further define the changes in the microtubules in our myectomy samples, our HEM samples, with and without sarcomere mutations. And we found that there's a high increase in alpha tubulin, in particular in the sarcomere positive samples. And there's a high increase in detyrosinated tubulin and also in the acetylated tubulin. And it's evident that all these changes are more severe in the sarcomere mutation positive group compared to the sarcomere mutation negative group. Now we wanted to link this with function of the cardiomyocytes, and there we used our HCM mouse model carrying the most frequent mutation, which is present in the Netherlands. It's a founder mutation in myosin binding protein C. And we did not observe a change in the level of alpha-tubulin, but we did find an enormously elevated detyrosinated level of tubulins in these mice. Then we went on to study cardiomyocyte function, and we took advantage of the system which is developed in collaboration with Cytocipher. So we can isolate single cardiac muscle cells with Langendorf perfusion, and we can measure multiple cells using the Cytocipher system. And using this system, we could see that in the mutant mice compared to the wild type mice, there's an impairment in the time to contraction, but also impairment of the relaxation. And if we do the repeated measurements after treatment with partenolide, which inhibits this detyrosination of the microtubules, we could restore contraction times and relaxation times of the HCM mice to the values observed in the wild type mice. So using, with this, we show that the detyrosination of tubulin, which is elevated in hypertrophic cardiomyopathy, may be a treatment target in hypertrophic cardiomyopathy. So to summarize the data thus far, we've shown that mutation location is really an important determinant of the change, at least in calcium sensitivity, and may be used as a risk predictor, but this has to be studied systematically in longitudinal studies in patient cohorts. Colleagues from Myocardia and the team of Jacopo Olivotto showed that mavacamten is a potential drug targeting the disordered ordered myosin state in the heart. And then studies of our own team show that detyrosination of tubulin is highly increased, in particular in mutation-positive hypertrophic cardiomyopathy, and maybe a promising new treatment target. And what we really need to put more efforts into is defining the main pathomechanisms in the sarcomere mutation negative patient group. This is really a large group among all HCM patients, and we should better understand what is the disease mechanism in this group to be able to give a proper treatment. So many people are involved in the studies I've shown during this presentation. I'm very happy to give this overview here. And thank you for your listening to my presentation. I'm happy to answer questions in the digital platform.